7C. Biological soil amendments and/or crop inputs that have gone through a validated treatment process, not including composting. In this video, we will review the metrics requirements for biological soil amendments and/or crop inputs that have gone through a validated treatment process, not including composting. These include requirements on their use and application, process validation, and their sampling and testing requirements, as well as any required documentation. Products. These products include soil amendments and crop inputs that have gone through a validated treatment process other than composting, such as chicken pellets, blood meal, bone meal, feather meal, soybean meal, kelp meal, alfalfa meal, cottonseed meal, mustard meal, rice bran, treated fish emulsion, and treated agricultural teas. Heat process validation. The metrics requires that any heat treatment processes used to treat soil amendments that contain animal manure must be validated to ensure that the process can reduce pathogens of human health significance to acceptable levels. Use and application interval. According to the LGMA metrics, if the heat treatment process used to inactivate human pathogens of significant public health for the soil amendment or crop input meets the following microbial acceptance criteria, then no time interval is needed between the application of the product and the harvest. But if the heat treatment process is not validated and is still likely to reduce microbial populations of human pathogens significantly, and it has a product certificate of analysis that meets the microbial acceptance criteria, then a 45-day interval between application and harvest is required. Nonetheless, remember that all soil amendment and crop input products need to be used in accordance with all local, state, and federal regulations. Target organisms and acceptance criteria. To ensure food safety, microbial testing of the product is required, meaning that these soil amendments and crop inputs must comply with the following acceptance criteria before they can be used. Fecal coliforms, negative or lower than the detection limit per gram. Salmonella, negative or lower than the detection limit, lower than 1 mpn per 30 grams. STEC, negative or lower than the detection limit per methodology used. Listeria monocytogenes, not detected or lower than the detection limit, less than 1 CFU per 5 grams. Sampling plan. To ensure proper microbial testing of soil amendments and crop inputs, a random sample representative of the material must be taken by a trained sampler and/or verified automated process. For solids, a minimum of 60 samples are required, while for liquids, the sample size needs to be per production process lot sizes. Testing frequency. You will need to test each soil amendment or crop input lot before it is applied to the production fields. In this case, the LGMA metrics defines a lot as a specific quantity of a finished product or other material that is intended to have a uniform character and quality within specified limits and is produced according to a single manufacturing order during the same cycle of manufacture. In addition, you will also need to test any product that you recondition or reprocess because it is suspected of being contaminated. Recommended test methods. When testing soil amendments and crop inputs for target organisms, make sure your lab uses the test methods recommended by the LGMA metrics. Documentation. For 7C products, documentation that describes the process validation for eliminating the pathogens of concern must be available. Finally, you are responsible for keeping all documentation and records that demonstrate that these soil amendments and crop inputs are free of pathogens of concern. This includes keeping any test results, certificates of analysis, and documentation current and available for verification for at least 2 years. Policies, procedures, letters of guarantee, and similar types of documents must be updated annually. The certificate of analysis for the product will also need to include lot information required to conduct traceability for the material applied to the growing location and to link the product to a test result. This information includes any data that could be used to confirm the lot description, such as the lot identification number associated with a treatment step, shift, time parameters, sanitation breaks, volume, weight, size, and any other parameters that could be used based on a specific production process. This information needs to be included on the certificate of analysis, or it must accompany the certificate of analysis if it cannot be described directly on the certificate. Lastly, if you process your own soil amendments and crop inputs, process control monitoring records must be reviewed, dated, and signed within a week after the records are made by a supervisor or responsible party. This is the end of our video on 7C, biological soil amendments and/or crop inputs that have gone through a validated treatment process, not including composting, developed by Food Safety CTS for California LGMA.